Hello, my name is Victoria Ortiz, and the focus of my project is the effect of stress, sleep, and a poor diet on cognitive performance. I chose this topic because teenagers are constantly trying to reduce stress due to their schoolwork, fitting in sleep after doing extracurriculars, and having little time to eat healthy, balanced meals. The first question I asked in my research process was, what exactly is cognitive performance? So cognitive performance can be defined as the ability to carry out specific mental or psychologically stimulating activities. It encompasses a variety of skills and abilities, including intellectual capacity, attention, concentration, and memory. For my project, I focused on cognitive performance in adolescents. The reason I became interested in this topic is because many everyday aspects of adolescent life impact cognitive performance. Some good habits and behaviors can improve cognitive performance, but there are many more that can decrease it, such as high stress, sleep deprivation, and a poor diet. Adolescents are expected to learn and perform their best academically while following a demanding schedule of extracurriculars. Several studies have already shown that my generation suffers from heightened levels of stress. In addition, a demanding schedule can lead to sleep deprivation, and the everyday running from one activity to the next decreases the time required to enjoy proper meals and maintain a healthy, balanced diet. I've observed that these last three factors apply directly to almost all of the high school students I know. Several studies have shown that stress, lack of sleep, and a poor diet can directly affect memory and retention, as well as attention in the classroom setting. My personal observations at school and background research I conducted led me to formulate a hypothesis. This ARP was aimed to study the effects of chronic stress, sleep deprivation, and a poor diet in cognitive performance in adolescents. So I'll split up my presentation into two parts. First is the cumulative information I gathered from reading literature, and the second part will be discussing the lab work we conducted on the correlation between leptin levels, body fat, and genes that promote obesity. So to further understand my understanding on stress, sleep, and diet, I read published studies on cognition in both animal and human models. Most of the sources I looked at were from PubMed journals, Nature, Neuroscience Reviews, and Science magazine articles. I also understood the data of experts in the field that have conducted experiments on each of my three factors. After this research, I found some similarities. Chronic stress decreases immunity and affects the physiological function of the brain. It also impairs memory in humans and rodents and decreases brain plasticity. When looking at chronic stress in adolescents, this can lead to school grades decreasing, which can also lead to more stress and completing the cycle. This can lead also to sleep deprivation if you're stressed out. So sleep deprivation can decrease problem-solving abilities, decrease retention, and decrease attention as well. A poor diet has a direct effect on the gut microbiome. So a poor diet, which is considered high in fats and added sugars, will increase body fat and therefore increase leptin release. So signaling to the hypothalamus with too much leptin leads to leptin resistance. This can affect cognitive performance. So therefore, poor diet does decrease cognitive performance. In addition, I also conducted lab research to study the effect of body fat and leptin. So leptin is a hormone that suppresses hunger and is produced by fat cells. So the more body fat you have, the higher your leptin levels are. My research focused on new genes involved in leptin release from adipocytes or fat cells. We studied a specific type of gene called OMS1. This gene is located at the base of the cilia in the cell, and the cilia, as shown here, is like an antenna their job is to send messages to other cells. OMS1 is involved in obesity because when deleted in mice, it induces a large increase in body fat. So this led us to form a hypothesis. We hypothesized that the deletion of the OMS1 gene would increase leptin release from fat pads of young OMS1 knockout rats. What we did was we took young and old rats and obtained abdominal fat pads from each. We then sectioned these pieces of fat evenly and incubated each piece for 2 hours. This allowed us to measure the leptin release. We then used a method called RNA sequencing to find the expression of all genes in the normal and the obese rats. So the body weight of both the OMS1 knockout rats, shown here, and the wild-type rats were measured over time, and the rats were placed on normal chow, which means They were given the same amount of food and the same type of food. We see that as the normal rats are growing, the alms 1 knockout rats develop obesity and gain much more weight than the normal rats. And you can see this difference, especially after 12 weeks. When looking at the leptin release, we measured leptin at 7 weeks before the rats were obese, at 12 weeks, and at 16 weeks. Importantly, at 7 weeks, we found that leptin levels for the alms 1 knockout rats were already 8-fold higher than the normal rats. Of course, after 12 weeks, the alms 1 knockout rats were hyperleptinemic, and this is maintained when the rats are obese at 16 weeks. To actually show that this defect is somewhat related to adipocytes, we took abdominal fat pads from the young, or 7-week-old, and the older, 16-week-old rats, and incubated the fat and measured the release of leptin to the media. What we found is that in the young rats, there was a very high leptin release that is much higher in the knockouts than in the wild types. In the older rats, the leptin release from the fat is less, but still the alms 1 knockout rats produce much more leptin than the normal or the wild type rats. In order to understand the alms 1 function and adipocytes, we isolated the same fat pads and ran RNA sequencing. This allowed us to measure the expression of about 16,000 genes in the adipocytes of the normal and the alms 1 knockout rats. What we find is that there are 234 downregulated genes, and these genes are involved in cell growth and differentiation. They also found 11 genes that are involved in ciliary pathways. And this is important because all the genes that depend on the 11 ciliary genes are also going to be downregulated. This data suggests that alms 1 somehow controls the expression of genes that depend on the ciliary genes. And this is me presenting my work at the American Society for Cell Biology meeting this past December 2019. From this, we conclude that alms 1 is likely involved in leptin release from adipocytes. Our data also points to an important role for alms 1 in regulating ciliary gene expression in adipocytes. So to tie both my lab work and my literature studies, published data shows that chronic stress increases leptin in the hypothalamus, and a diet with increased fat and sugar also increases leptin. Sleep deprivation actually decreases leptin levels, leading to a feeling of hunger. This is important because it acknowledges my work on stress, sleep, and diet, as well as the research I conducted on leptin. Lastly, I want to acknowledge a couple of people that have helped me along the way during my ARP process. First is Dr. Emilio Motillo, and he was my advisor during the leptin research project at Henry Ford Hospital. Next is Ms. Mwala for reading over my work multiple times and answering any questions I had. Next is the Ortiz and Mendez laboratories for the space and assistance and offering to help me while I was conducting my research. And lastly, of course, my parents for provoking my curiosity and answering any questions I had. Thank you so much. I hope you enjoyed this, and please feel free to reach out if you have any questions. Thank you.